All right. Excuse me. Time to get started. Good morning, everyone. Today, this is the first webinar session dedicated to young scientists as speakers. And actually, Netpore wish to encourage PhD students and postdoc to present their activity, to present themselves and their research work, and eventually to build connection through this monthly webinar that this webinar meeting we have now for about a year and a half in Netpore. So today, it is my pleasure to introduce the first two young researchers of Netpore webinars. We'll be starting with Mirjana Monstig. Good morning, Mirjana.
And thank you for
Mirjana has
your
a
kind
bachelor and
invitation.
master's degree in biochemistry, and she's currently a PhD student of biochemistry at the Faculty of Sciences in Novi Sad in Serbia under the supervision of Nikola Knežević. And she's also a junior research assistant at the Biosense Institute in Serbia. Mirjana is working on the synthesis, functionalization, and characterization of silica-based materials and their application in cancer drug delivery and MRI imaging. And today, Mirjana will be talking about mesoporous silica nanoparticles, also called MSN nanoparticles for glioblastoma, multiform theranostics. Please, Mirjana.
Good morning, everyone. Thank you for the kind invitation and thank you for this introduction. So please let me share my screen once again. I think it should work. Is it okay?
Yes, it is.
Okay, great. Thank you. So my today's topic, and actually the topic that our research group is working on, is mesoporous silica nanoparticles. We are especially working on its usage for the treatment of glioblastoma multiforme. And to be more precise, we are interested in theranostics. Theranostics is a, let's say, new word which comes from the combination of therapy and diagnostics. So we are looking to find the way to deploy mesoporous silica nanoparticles both for therapy and for the diagnostics of glioblastoma multiforme. Glioblastoma multiforme is very, very bad type of brain cancer. So let's start with one short overview of the conventional modalities for cancer treatment. First of all, we all know about the surgical intervention. We know about the radiotherapy and chemotherapeutic drugs, or the combined therapy, the therapy that combines both of them, radiotherapy and chemotherapeutic drugs. What happens is that there is actually the rapid drug metabolism, and there are a lot of harmful side effects when those kind of conventional modalities are being used. And nowadays the drug resistance is also high, so the drug uptake is actually reduced and the drug efflux is increased. The worst thing that happens by using those conventional modalities for cancer treatment is that there is a huge damage of actually healthy cells, so a high toxicity to patient is monitored. Because of that, nanomedicine is a hot topic these days, and nanomedicine can be defined as a branch of medicine that seeks to apply the nanotechnology. So it is the combination of nanotechnology for the purposes of cancer treatment. Anyway, the main goal is to safely deliver the drug to the specific parts of the body and to release the drug at that specific site where it's needed without causing the harmful side effects. And what is also important is that we want to be sure that we can accurately detect how the treatment is working in this specific area. Because of that, a lot of groups around the world are nowadays actually synthesizing nanoparticles and talking about their application in cancer treatment. First of all, the usage of nanoparticles as the drug molecules carriers actually protects the drug molecules from the premature degradation, which is very important. And it also prevents the drugs from the prematurely interacting with the biological environment, which actually means that their toxicity is minimized. The intracellular penetration can be improved in this manner of using nanoparticles as the drug delivery system. And there is the possibility for passive and for active targeting. So passive targeting is the accumulation of those nanoparticles loaded with different dyes, drugs, or contrast agents. And those nanoparticles actually accumulate at the targeted site because of their surface charge or their shape or their size. But what is more interesting is the active targeting. Actually, that is the possibility for the modification of nanoparticles with, for example, aptamers, antibodies, or peptides. And in that manner, they can recognize and bind to the receptors or the biomarkers that are overexpressed on the targeting cells or tissues. In that way, those nanoparticles can, let's say, spend more time at that specific site, and in that manner release more of the cargo, actually more of the drug at the specific site. To be sure that we selected the suitable nanoparticle, we have to make sure that the material and the drug that we want to deliver are chemically inert to each other so that they don't interact in between. The nanoparticles should have the high drug loading capacity, which is important so that they can carry the suitable amount of the drug, and they should have the good targeting capabilities and the possibility to release the drug in a controlled manner. Of course, as everything, they should be easy to obtain, they should be cheap, they should be biodegradable, they should be stable, actually easy to functionalize, and well explored. What is also important, and I would like to stress out, is that the nanoparticles should actually show the low rate of aggregation, because we need the extended circulating time, and in the best way possible, that there should actually be no rate of aggregation. Speaking of cancer and speaking of nanoparticles, I cannot miss to talk a little bit about the enhanced permeability and retention effect. Actually, both of these things are very important, because enhanced permeability actually means that tumor blood vessels are often characterized by irregular and leaky structures, and that actually allows for the nanoparticles to cross the wall of the cell and actually of the cancer tissue, and to spend more time inside the circulation system. And retention effect is caused by the tumors that often lack an efficient lymphatic drainage system, which means that the longer retention of those nanoparticles can be achieved, and because of that, the longer release of drugs into tumor cells. Mesoporous silica nanoparticles and the method for their synthesis is actually known since 1992, as shown in this small picture. Everything starts with the surfactant molecules being dispersed in a solvent. So the surfactant micelles are made in that manner, and the polar heads, they actually turn to the outer part, and the inside part is filled with the hydrophobic tails, let's say it like that. Then they form the micellar rod in a hexagonal array, and after the addition of silicate, or of precursor as we call it, silicate species actually occupy the space between the cylinders, and in that way we have the structure shown in this part of the picture. And after the calcination, or after the washing out of those organic groups, we actually get the MCM41, that is the name of that structure, which has a hexagonal array, and whose pores are actually empty and ready to accept drug, dye, contrast agent, or whatever is needed. MSNs has some great characteristics to be used as the system for bringing nanoparticles, for bringing the drugs to some specific area, because it has a high surface area. So a selective functionalization of external surface or on the inside pores can be achieved. The diameters of the pores is only from 1 to 50 nanometers, which is great. And they are chemically inert, they show high thermal stability, they are biosafety, and they have a possibility to entrap the molecules and actually to trap them and onto a signal, which can be the change of pH value, the change of temperature of some chemical triggers or something similar. They can trigger the cargo release. This is the way we synthesize our nanoparticles in our lab. So it is the surfactant templated and base catalyzed co-condensation of silica precursors. Everything starts when we actually mix acetyl 3-methylammonium bromide in water, and we add potassium hydroxide. And then we add tetraethoxysilane, which is a precursor, and in that manner we form the nanoparticles, which should be washed out from the surfactant, which is inside of those tubes, let's say like that, and we usually do it in the presence of acid and methanol. This is the picture that we usually put inside of our project proposals, or when we want to be part of someone else's project. So this picture summarizes all of the research possibilities of our nanoparticles. So this blue ball, let's call it like that, is a porous nanoparticle. And the black one the black one can or doesn't have to be here. This is a magnetic nanoparticle used as a core. But what is important is this darker blue entrapped therapeutic or signaling molecule inside of the pores. Probably the most important in this picture is the red balls, because those are the molecules that are blocking the pores. So let's say it's more easy to fill the pores, to load the pores, but it's very important to prevent the drugs, for example, from leaking out of the pores before the right time comes. Here we also showed the surface molecules for interaction with the tissue. And this green shows the possibility for additional functionalization for improving interaction, or for actually increasing the solubility. It is also important in some cases. Coming back again to the picture that we saw a bit earlier, because I want here to show that the shape of the nanoparticle is really important, because according to the literature, the spherical nanoparticles have a minimal lateral drift, and they're less likely to marginate to vessel walls, which means longer life, longer circulation lifetime, in comparison to the non-spherical nanoparticles. So it is important that we also think about the shape of the nanoparticle and what we want it to do when it enters the biosystem. So here is actually the difference evident. So the non-spherical ones bind to the vessel walls, and the spherical ones, they have a longer circulation time, and because of that, the longer time of releasing the drug at the specific area which we want to achieve. Also important is the surface charge. So again, according to the literature, the positively charged nanoparticles are more prone to, let's say, be eaten by macrophages in the lungs, liver, and spleen in the comparison to the negatively charged or the non-charged nanoparticles. Okay, so here is the system. We call it the pH-responsive cyclodextrin-capped MSNs for cargo delivery. So I will go step by step to try to explain our scheme. So this is the MSN nanoparticle whose pores are filled, in this case, with the ruthenium dye. So it is just the first experiment what we wanted to see, whether we can load the pores with the dye and whether we can release it upon the lowering of the pH value. So this is a hydroxide bond, and upon the lowering of the pH value, it should be cleaved. That actually has happened. So here are the results of the release kinetics for the ruthenium dye. We perform these experiments in vials. So we actually disperse the material in the buffers of different pH values, so pH 5, 6, and pH 7.4, as shown here in different colors. And then we stir that material and then centrifuge it after certain points of time. And then we measure the absorbance of the supernatant. After the measurement, we return the supernatant to the material, we vortex it, and then we continue with the stirring till the next time point. So this is what we got. This was up until 140 hours. And when we normalize the release kinetics, here we can also see that the release is lower for the pH 7.4, but when we lowered the pH value up until down until the 5 p.m., the release kinetics is higher, which is good because we all know that in the presence of cancer cells and cancer tissue, the pH value is always lower. So this is a good trigger for the releasing of, in this case, ruthenium dye. Here we also used the ruthenium dye, but this is the redox responsive system. So it's not the lowering of the pH value, but the addition of glutathione. So the cargo is again ruthenium dye, and here we can see the absorbance of the ruthenium dye, as we previously said. But here is shown that the addition of glutathione, it actually the absorbance before the addition is around 0.04, and after the addition, this is of 10 millimolar glutathione, the addition goes up to, the absorbance, goes up to 0.15. Here is pretty much the similar result. We just wanted to add the glutathione in portions, so we got those slopes, but the effect is the same. So this is the proof that we can load the pores, and then upon the addition of glutathione as a trigger, we can release the ruthenium dye in this case. And actually, as the lowering of the pH value, glutathione is also measured in higher concentrations around the cancer tissues and cancer cells. The system that we are working on still and that we have some manuscripts submitted is this pH responsive targeted diagnostics of glioblastoma multiforme. So the principle is the similar. We actually, in this case, loaded the paclitaxel, that is the anti-cancer drugs. It can be doxorubicin, it can be temozolomide, there are different kinds of those drugs. We actually chose paclitaxel for these purposes. And what is important is that we have chlorotoxin. Chlorotoxin is targeting cancer overexpressed protein MMP2. So MMP2 is matrix metalloproteinase 2, and it is overexpressed in the cancer cells. And this chlorotoxin can target it, so it binds more specifically to the cancer cells, to the glioblastoma in our case, to those cells. Please note that those results are from the manuscript that is submitted, but anyways, I wanted to show what is the latest from our lab. So we actually started with, in this case, mercaptopropyl functionalized MSN, and then functionalize it with the group that has maleimide group, and in that way we have here the thioether linkage. This is the system that we will from now on call functionalized MSN. This functionalized MSN gets loaded with gadobutrol, which is a contrast agent, or with the anti-cancer drug paclitaxel. And because there is a hydrazine-based molecule, I mean here is the residue, there is a pH-sensitive hydrazone bond in this loaded and kept MSN, as we will refer to it from now on. So we have a functionalized MSN, then we have it loaded and kept with beta-cyclodextrin. And then when we actually add that chlorotoxin, the targeting molecule, which is attached covalently to the adamantane amine, it just further utilized the strong host-guest interaction between the adamantane amine and the chlorodextrin. So this is a targeting MSN. To begin with some results, those are the SAM and TAM micrographs from the MSN, these two, and this is from the functionalized MSN, the first one that we modified, so it is the functionalized MSN. From both SAM micrographs, we can see that the particles are spherical and that the diameter is from 100 up to 250 nanometers, which is ideal. And from the TAM pictures, especially from this one, we can see that those particles are porous. Actually, XRD measurements followed the SAM and TAM measurements. And what is nice here is that both MSNs and functionalized MSN exhibit the 2D hexagonal pore structure as we wanted to achieve. And what is also good is that the functionalization did not significantly influence the pore structure. So we have pretty much the same peak. To be sure that we performed the functionalization good, we were looking for the differences between the Fourier transform infrared spectroscopy spectra actually, between the MSN and between the functionalized MSN, and we found it here. So these vibrations at around 1700 are ascribed to the vibrations of the carbonyl groups. So now that we were sure that the material was good, another thing was performed. It was a BET and BG8 characteristics of the starting material. As expected, both of them have that shape have that type 4, which is actually expected. And the thing is that after the functionalization of MSN, the surface area decreased, and actually the total pore volume also was lower. What is nice and good is that the average pore size remained for 2.4 nanometers, and the predominant BGH peak can be seen in both cases at 1.9 nanometers. Here are the calibration curves. This should be a supplemental information, but these are the calibration curves for paclitaxel, for that anti-cancer drug that we actually loaded. So we have it at pH 5, 6, 7, and also in water. And this is the part that I'm mostly interested in, since I'm a biochemist. We actually performed the viability tests. So to begin with, we performed the viability tests to check the toxicity on U87 glioblastoma multiforme cells. We performed the CCK8 using the CCK8 test. So it is a spectrophotometric method similar to MTT test that is probably more widely used. Anyhow, we have the results after 24, 48, and 72 hours of incubation. And in all three cases, the functionalized MSN showed no toxicity, or low toxicity, only in this case of 100 micrograms per milliliter, but those are so high concentrations that they're usually not even used in those high concentrations. We have here, green is the anti-cancer drug loaded and capped material. And the gray one is the same as the blue one, but with the addition of that targeting chlorotoxin molecule. So the results are not actually the same, especially after 42 hours. But the thing is that the loading capacity of paclitaxel drug non-targeting is actually much lower than the capacity of this one target with the targeting molecule. So we can ascribe this higher, let's say, the lower viability of the cells maybe to that characteristic that there is more of anti-cancer drug loaded in this gray material. Working on the more or less same principle, we have a pH responsive release of contrast agent. So this was all about the anti-cancer drug, and this is the same system but now loaded with the contrast agent. So here is the result of the release of contrast agent during the time, as you can see, up to 24 hours. But the thing is that the highest release is observed for all three different pH values after, let's say, it was around 30 minutes. So it is a rapid release, more or less, but what we think has happened is that after we put back the supernatant that we have just measured, so the first measurement is usually around 30 minutes, the contrast agent might be absorbed on the surface of the nanoparticles, and that's why we see this high peak at 30 minutes. But anyways, what we also here proved is that the release is the highest when the pH value of the buffer is the lowest. So it is a good proof of what we wanted to show here. As expected, the BET isotherms have changed their form. So they are now not type 4, but type 2, which is actually expected for the beta-cyclodextrin capped material. And what I wanted to stress out here is the amount of loaded drug or loaded contrast agent. So contrast agent, the one that we use, is gadobutrol, but its molecular weight is much lower than the molecular weight of the paclitaxel. So the amount of the loaded contrast agent is twice or even 3 times or more higher than the possibility of this material to get loaded with the paclitaxel, which is also important to have in mind when thinking of which drug or which contrast agent we will use for our experiments. Here are similar as for the paclitaxel. Here are the calibration curves for the contrast agent at different pH values, again 5, 6, and 7.4. And my favorite part, the toxicity on U87 glioblastoma multiforme cells. What we have here is that the viability is pretty much the same for both of them, for both the materials. The material loaded with contrast agent and only kept, or the material loaded with contrast agent kept and with the addition of chlorotoxin, or I mean the addition of the targeting agents. We are speaking about contrast agents. So the main idea behind the loading of contrast agent is to see whether we can track the release of it from the mesopores upon the exposure to weakly acidic environments. Because of that, we performed magnetic resonance imaging, and here are the results. Here are the pseudocolor MRI images. We actually used the plain MSN, like the starting material, and we used the material loaded with contrast agent and kept. So here is no chlorotoxin, but never mind. This was all after 24 hours, and after 48 hours, we took the supernatants. And here are the results. So I think these ones are the easiest to read. there is, as we can see, the significant difference between these two colors, even for pH 5 or 7.4, and the results that we obtain for the MSN. These are yellow, I mean around 3. This is the relaxation time, T1 time, and these ones are lower. The lower the relaxation time, the higher the release of contrast agent, which means that the relaxation is higher as it goes more to the green on this scale. So the relaxation, so the release of contrast agent is higher at the pH 5 than it was to the pH 7.4. But I know it is pretty hard to interpret these results. That's why we have here not so nicely colored, but these are the results. We actually took the region of interest. Why did we do that? I have to go back because I forgot to tell you. So this is for example, this is the sample, and this is the aggregate of the mesoporous silica nanoparticles. Over time, they do tend to aggregate, and the thing is that they aggregate at the bottom of this cuvette that we used for the MRI measurements. So this is actually the precipitate, and this is the aggregate, let's call it like that. So here are the results for the MSN material and the results for the contrast agent capped materials over time. And we took the region of interest. It is the same, let's say, region here, and we have it for the bottom and for the top part of the cuvette. And here are the results, I mean in numbers, the time constants, which determines the rate at which excited protons return to equilibrium. It is known as the T1 values. When we put all of the results in a form of graph, this is what we get. So we have the results for the bulk solution, and we have the results for the precipitate. For the precipitate, we actually did not have any difference, let's say, between the more or less, between those samples. Gray and yellow are the MSNs without contrast agents, so they have a higher T1 values because there was no release of contrast agent. Here, the contrast agents were released, but more or less their relaxation time is the same. Because of that, we actually have the results also for the bulk solution. There is difference. It's not that high, but it can be seen. So the longitudinal relaxation time is lower, a little bit lower, but lower for the material at the pH 5. Anyway, very important adjustments are needed for the further applications in real settings. And what we are looking for is the significantly higher relaxivities of contrast agents. Because of that, we will work later on, and we will continue working on the synthesis of the MSNs with a higher release capacity. And we should definitely improve the sensitivity of MRI measurements in the future. This is again some, let's say, biological stuff, but I think it's really important. These are the results from the confocal microscopy when we labeled the nanoparticles with fluorescein isothiocyanate. So here are the FITSI labeled MSNs. These ones are the same, but they are kept with the cyclodextrin, and these ones have the additional chlorotoxins. So these ones, let's say the plain ones, the ones that are kept, and the ones that also have the targeting agents. Red is the cell membrane, blue is the nuclei, but what we are looking for is this green color. So this is the proof that the nanoparticles actually can enter the glioblastoma multiforme cells. What we also wanted to make sure here is that none of those material is toxic to the cells. So neither only kept, neither the targeting and kept material is toxic to any of these cells. Anyway, this confocal microscopy only shows us, proves us that the nanoparticles do enter, but we don't know nothing about the amounts. And maybe this will This wasn't that much expected, but it is as it is. So flow cytometry histograms for those nanoparticles, again, the same three ones. So these are the, let's say, plain ones, the capped ones, and the ones with the targeting agents. And here is probably much easier to see that the highest, let's say, the best internalized ones are the capped ones, which is strange because we expected these ones with a targeting agent to be best internalized. So what has happened, if we go back to this picture, I mean, we can see them, but the thing is that with the flow cytometry, they are not being internalized. They are actually interacting with the MMP2, which is overexpressed at the membrane. So it prevents them from entering the cells, but it actually prolongs their time and their actually amount on the surface, on the cell wall of the nanoparticles. So it can be actually interesting if the time that they spent close to the nanoparticles is long enough, maybe they can release more of the drug, and as we saw, they do kill more of the cells. So they have a different mechanism for doing that in comparison to these only kept nanoparticles that can enter the cells better. So to conclude everything that I said so far, since everything is being in a form of manuscript, but we are waiting for the exception. But let's say we showed that starting materials are biocompatible, which is important. And we demonstrated that the contrast agent and that the anti-cancer drugs can be loaded into the pores and can be released in acidic environment, and in one case we showed also with the addition of glutathione. What is also important is that when we actually add chlorotoxin, the cancer targeting ligands, we get the higher possibility to kill the cancer cells, which is as we expected, although we are not still sure about the mechanism in which it happens, because flow cytometry shows that those targeting nanoparticles don't enter the cells. And the preliminary experiments that we did on MRI we did on MRI actually confirmed the capability to identify enhanced release of contrast agents under weakly acidic environment. But our research is ongoing, and we are looking for the MRI-based evaluation of nanomaterials for the targeted drug delivery to specific tissues, with a hope that it can en route to a more personalized treatment of cancer one day. So ideally, we would one day have an opportunity to check the release of contrast agent using the MRI imaging, and then upon that, it can go to a more personalized treatment of a cancer and using the drugs that are more suitable to that patient himself. And for the end, I would like to thank you for the invitation once again, and to thank my mentor, my professor, Dr. Nikola Knežević, also my colleagues Minja Mladenović and Aleksandra Pavlović. And nothing of this would be possible without the Biosense Institute here in Novi Sad and these two projects. So we have NanoFacts, the twinning project, and we have the project from the Science Fund of Republic of Serbia. It all allowed us to have a great collaborators. So we are collaborating with the Trinity College in Dublin, with the Max Planck Institute and Dr. Jelena Lazovic. And actually I spent some time in Wageningen University to perform the MRI experiments in the lab of Professor Aldrich Velders. And thank you all for listening and coming to this such nice event. Yes.
All right, can you hear me? Okay, thanks guys. I have had trouble with connection. Thank you very much, Marina, for your great talk and excellent results. Very, very interesting approach for drug delivery.
Mm.
We have a very short time for question. The talk was a bit long, but that's very interesting. So please, someone, if you have a question for Mariana, please go ahead.
Can we ask some question? I could not understand you completely.
I also have some problems with internet, but if we are okay with the time, I'm here to answer.
Yeah, in principle, these particles which you synthesized are very interesting, also for some other applications. And there's a question, maybe we can have some samples, also not loaded ones, but the unloaded ones, because we are working with such particles, but usually we have not non-porous silica particles, and if we have porous ones in the same size range, then that would be interesting to load them and to use them as well.
Of course, why not? Yeah, we always have them unloaded just to check whether they are themselves probably toxic or not. But all of the experiments that we performed show that they are non-toxic, and the pores are really big, so that they can entrap a lot of contrasting, a lot of different kind of agents. So yeah.
Okay, then I would write you, and then maybe you can send
Thank
us
you. Thanks a
something.
lot. You can send it to my email address or my mentors, but of course, we are looking for some collaborations. Why not?
Okay, nice. And maybe a short second question. If you are applying such silica particles, you have not only the toxic problem, but you have also the bioclearance. Also, if you have these particles in the body, they have
Yes.
to be cleared after a while. Is that working, or is there no solution yet?
To be honest, this is a tough question. I'm at the moment waiting for my visa to go to the Trinity College Dublin and to continue my experiments in vivo. So if you ask me in few months, I will have a better answer. Looking to the literature, the clearance actually goes through the liver mostly, and they shouldn't be toxic in that case. But since we are working on the glioblastoma multiforme, it is one of the most, let's say, difficult types of tumor because it is the brain and everything. So yeah, we are definitely going to work in vivo because all of this cannot go to some later stages of preclinical trial before we do it on the animals. So yeah, this autumn hopefully we will have some also in vivo results.
Okay, thank you.
Thank you. Thank you.
Okay, thank you very much. Thanks a lot, Lars, for the great and interesting question to Mariana. I'm sorry, Mariana, because we have to switch to the next
Of
talk.
course, yeah, sorry
This
for taking the
morning,
time. It's my
we have
first time doing this, so
it was very good, very clear, and very interesting. You're very good
thank
at
you. Thanks
talking.
once again.
So now I'm
Okay.
happy, very delighted to introduce the second speaker of the morning, who is Ofer Prince Setter. He's currently in his last year PhD at the Faculty of Biotechnology and Food Engineering at the Technion Institute in Israel. He works under the supervision of Professor Esther Siegel at Technion, and his research focuses on between nanotechnology and microbiology. It's at the interface between both. Prior to his PhD, Offer received his bachelor in biotechnology and food engineering from Technion as well. It was in 2012. Then he spent 5 years at Teva Pharmaceutical Industry, mainly coordinating drug product analysis, which I believe has been a very enriching and interesting experience before going to a PhD program. It's probably a very important competence, additional competence that you have for for doing research now. He's also currently a fellow of the prestigious Israeli Foundation, and also has already very exciting plans for his coming postdoc in Professor Kisling Lab at UC Berkeley. So today he will be talking about the good China, very interesting, intrinsically mesoporous nanoclay for the selective delivery on antimicrobials, avoiding these biases? Please offer. It's your time.
Okay, thank you very much for the kind introduction. Let me share my screen. Okay. Okay. Okay, due to the shortage of time, I'll try to be brief, but please stop me if I go beyond the allotted time. Okay, thank you everyone for attending this seminar. So my name is Ofer Penseter, and I will try today to convince you that there is a connection between porcelain and antibiotic treatment. So in our lab, I think probably most of you know, the Sega lab, we are working mostly on porous silicon, but my work focuses on another material, natural clay, which is also porous and siliceous. The motivation for my work stems from antibiotic-induced dysbiosis. When we take antibiotics, we might also harm the commensal beneficial populations of bacteria in our body, and these mostly affect the gut, leading to the undesired condition of dysbiosis. So most of you will think, okay, this is just some abdominal discomfort, but actually current studies show that this condition relates to plethora of illnesses and conditions, even affecting the brain and the heart. Some of them may emerge years after taking the antibiotics, and it takes a lot of time for the microbiome to retain itself, and the burdens onto the authorities of health may be dire. So one approach to overcome this challenge is to use carriers, nanocarriers, to deliver antimicrobials directly to the target bacteria. And of course, we've heard the wonderful talk about mesoporous silica and other nanomaterials are proposed. However, the synthesis of these synthetic options could be complex, and they might be too expensive for the antibiotic market. They are good for treating cancer, but for antibiotic treatment, it could not be a good option. So we try a completely different approach. Why not use completely natural materials? And here is an example for nanoclay. This is a mined mineral that exhibits nanomorphology, and we aim to functionalize it with capture probes that it would selectively target the bacteria and release antimicrobial cargo at their proximity. So I'll briefly go through the steps I will talk about in this talk. We'll start discussing the loading of the antimicrobial cargo, which is ciprofloxacin in our case, onto the clay. And then I'll discuss the synthetic route of immobilizing antibodies onto this material. Combining these two, I will discuss the antimicrobial effect and whether it is indeed as selective as we engineered it to be. And finally, I will go through a study, very interesting study that we performed using real human microbiome to see how this system actually works in a real life scenario. So starting with our raw material, this is clay. Most of you know porcelain, but actually under very close observation, we could find very intriguing nanomorphologies, completely natural, from rods to tubes and flakes, and are ideal for loading. And our material hallocyte nanotubes, even some of them are even approved for topic formulations. And our material hallocyte nanotubes is the tubular nanoclay. They are found all around the world. They are abundant. They are made of alternating layers of silica and alumina rolled into these tubes. Here you can see the lumen. And they are extremely low cost in comparison to other nanomaterials. They are bought by the kilogram, only a few dollars. It is very tempting to identify them as biocompatible, but actually, according to a survey we performed recently comparing hallocyte nanotubes to other synthetically commonly used nanomaterials, such as multi-wall carbon nanotubes, we saw that the picture is a little bit more complex. This is some of the threshold we found. So we can conclude that at least hallocytes are not more toxic than other nanomaterials. And these all attributes of hallocyte nanotubes render them ideal for all kinds of biomedical applications, such as tissue engineering and drug delivery, all based on their interactions with biological cells, starting with the very basic colloidal forces and then going through more complex interactions, as we heard in the previous talk about internalizing. In the case of bacteria, there could be adsorption on the bacterial cell and even incorporation with the biofilm. So our aim in this project is to direct these interactions of HNTs, halocyte nanotubes, with bacteria to be more selective using a capture probe. So the first step is the loading of the antimicrobials, antibiotics onto the clay. We are not the first one to do that. Actually, clays are very famous to be an adsorbent for pollutions of antibiotics in water. But we wanted to enhance this. And how? By acidic etching. If we perform this acidic etching on the halocyte nanotubes, we solubilize the alumina component, leaving a much more porous material, starting with 25 meter per gram. We can go up to 5 more times surface area, while fifth of it is indicated as a porous surface area. So the material becomes much more porous, which is very good for loading and release. We tried a variety of loading conditions, loading time, and concentrations of antibiotics. And at the constraints of antibiotic solubilization, we reached around 2% weight of loading. And interestingly, when we used pristine HNTs, which were not etched at the same conditions, 40% less loading capacity was observed. So indeed, the etching process enhanced the loading of the antibiotics. The next step is the immobilization of the antibodies. And on this slide, I will go step by step through the chemical route when FTIR, relevant FTIR spectra are presented on the left, starting with the HNTs. After the etching, and we expose additional hydroxyl groups, which are then aminoselenized using propylamino-trioxysilane. And then the amine groups are carboxylated. This is the amide bond that was newly formed. And using the very reactive intermediate, NHS intermediate, we conjugated protein A onto the surface of the etched clay. And protein A served as an anchor for the antibodies due to the affinity between the Fc region and protein A. So the antibodies are properly oriented. Used immunolabeling to localize the antibodies using a secondary antibody conjugated to gold nanoparticles, and you can see that the immobilized antibodies are evenly spaced on the clay nanotube. But are they active? For that, we used flow cytometry, but not ordinary flow cytometry. This time we used imaging flow cytometry that produces thousands of images of suspended objects. We take the fluorescent one. We did a mixture of the target bacteria, which is fluorescently labeled, and the modified clay. So we took only the images that has fluorescence with them and tried to use image analysis to differentiate between bound bacteria that are irregular in shape and the oval bacteria. So we have a plot of object area against object perimeter, and we found that the images depicting bound bacteria are found mostly in this region. So we use this region for all cytograms, for antibody-modified clay, non-modified clay, and bacteria. Of course, in all of these mixtures, we have bacteria, and we just counted the images that are in this region. The results are here normalized to the sample with non-modified clay, and we could see that the 2.5 higher affinity of the antibody-modified clay was observed against the target bacteria. A similar trend was also observed in a heterogeneous culture. Here we used 3 types of bacteria. Only one of them was the target bacteria. Each bacteria species is labeled in a different color, so we can differentiate between them. And again, we counted the images according to the image analysis, and we saw 4 to 2-fold higher occurrence of binding events for the target bacteria in comparison to the non-target bacteria. So here we have a system which is clay-based that could target bacteria in a culture. Under the microscope, aggregates of clay and target bacteria could be seen, and under higher magnification using electron microscopy, we could clearly see the bacterial cell attached to the aggregate of clay. Here you can see the tubes with the mouth of the tube, just ready to release antibiotics, but there is still no antibiotics in this system. And this brings us to the next step of combining antibiotics loading and immobilization of antibodies. So we have the loaded system, the multifunctional clay. It exhibited a sustained release profile in comparison to pure diffusion for the diffusion of pure antibiotics. And to assess its activity, again, we used, the loading of antibiotics did not impair the selective binding. Here again, it's a flow cytometry experiment, and you can see that the results for clay with antibiotics or without antibiotics is quite the same. So to investigate the antimicrobial effects of this loaded clay, again, we use the heterogeneous culture. The target bacteria are E. coli, the antibodies are against E. coli, and two other non-target species on the same heterogeneous culture. They are incubated with the multifunctional clay for 2 hours, and then the culture is spread onto 3 selective media, so we can enumerate each of the strains separately. And the results are here summarized as an equivalent ciprofloxacin concentration for clarity. Here you can see that for a range of particles, we used between 0.7 to 0.09 milligram per milliliter. For a range of concentration of particles, there is a clear better selectivity, enhanced selectivity for the target bacteria than the non-target bacteria, meaning that in the same heterogeneous culture, the clay actually exert a more intense antibacterial effect against the target bacteria than the non-target bacteria. And the assumed mechanism is that first the clay would bind to the bacteria, and due to the fact that the release of the antibiotics is sustained, most of it will actually be released at the proximity of the target bacteria, while the harm to the non-target species is reduced. In the future, maybe this kind of formulation could be used to treat gastroenteritis where the protection of the microbiome of the gut is very important, as we said earlier. To investigate this, we used human microbiome. We took from 5 donors, and we utilized a special system for fermenting feces under conditions that mimic the gut. This was developed by Professor Uri Lesmes from our department, and we did this in collaboration with his lab. These fermenters maintained anaerobic conditions, controlled temperature, and a very tight pH range. And we used the feces samples with 4 treatments. We used our multifunctional clay with antibiotics, only antibiotics, pristine clay, and only medium control. The fermentation went on for 24 hours, was sampled, and then we extracted the bacterial DNA. And according to the 16S RNA gene, we did the profiling of the microbiome. So I'll do only a very short summary of these results because these kind of experiments give a lot of results. So here you can see a heat map showing the abundance of bacterial taxa, all normalized to the medium control. So we can only see the differences between the treatments and the medium control. For blue is the multifunctional clay, red is only antibiotics, and green is the pristine clay along the fermentation time. So you can see that for most taxa, there's no much of a difference over time, except Enterobacteriaceae, which is actually the family which is most susceptible to ciprofloxacin. And in the literature, we have a lot of data about how ciprofloxacin is harmful to this very important family. But going into the genus level, into this family, we can see that our loaded clay also inhibited, but the target genus, Escherichia, in this family. However, the ciprofloxacin indiscriminately harmed and inhibited the entire family. And this is actually the first indication, and we can also see it using beta diversity. Here, the difference between the populations is expressed as geometric distances. So as the dots are more distant from each other, they are more different, and you could see that the clay loaded with antibiotics is very similar to the medium control and the pristine clay. However, the profiles of microbiome treated with pure cephalosporin shifts away, which means there is some disturbance of the microbiome here. And this is the first indication for the potential of natural clay modified with antibodies and loaded with antibiotics to exert a selective antimicrobial effect, protecting the microbiome, but still maintaining the potency of the loaded antibiotics. So to summarize this talk, I hope I gave you some new information about a new nanomaterial, which is completely natural, hallow-side nanotubes. Under acidic etching, the loading capacity is enhanced. We can modify them with antibodies to realize selective binding. Combined together, this multifunctional clay could exert an selective antimicrobial effect in a heterogeneous culture, and also in the human microbiome. Maybe in the future, this would be new formulations to take antibiotics without harming our precious microbiome. And for further reading, I welcome you to our former publications on which this presentation is based. And with that, I would like to acknowledge my group members, my professor, of course, Professor Esti Selagal, and the undergraduate who helped the Azrieli Foundation for the wonderful support, financial support, the Technion, and of course, you for your attention. And one last remark, if you are interested in more or working on nanoporous material for controlled release, I welcome you to submit this method collection I co-edit on Jove. So thank you very much.
Thank you very much, Ofer, for this great talk and presentation of a very interesting material for this application. I know your lab and is working a lot on this antibacterial project. It's always very interesting. So we have time, actually. Thanks for a few questions. If any of you have a question for Ofer, please go ahead. Okay, so I start with
I can ask one question.
Oh,
Yes,
please
please.
go ahead,
Yeah.
Alexander. Yeah.
So, nanoclay is mainly of alumina, is it right?
It is silica, alumina, phyllosilica.
Silica,
It's made
alumina.
of layers of silica and alumina. This is the basic, but there are more variations. There could be
So,
magnesium.
yeah, aluminum in general is not a biocompatible material. So, what actually happened in the body when you insert it in the body? And this is one of the questions that I ask, because we try to eliminate aluminum, to use aluminum for body, and this is this is may restrict you in a medical application in general.
Yes. So my answer is divided into two. So first, we did have some compatibility assays using CACO2 cells derived from intestinal epithelium, and no cytotoxicity was observed after 40 hours of exposure. And additionally, we use etched HNT hallocytes. So actually, there's almost no residue of aluminum. You can see it here with elemental mapping. Oh, okay. So here you can see a very small amount of aluminum retained in the etched hallocyte. And in general, the hallocyte nanotubes do not solubilize. They are not biodegradable. They retain as they are. And there are no enzymes that could degrade them. So most of the aluminum just kept as aluminum.
Yeah. Okay. Regards to ST.
Thank you.
All right. Can you hear me?
Yes.
Okay, sorry. Okay. Yeah. To complete on the question, I was wondering, Offer, if you had a chance to test the stability of the clays, if it's very stable over time, if it degrades quickly. This is very it's a real question. I have no idea. And if you had a chance to compare the stability of the pristine clay versus functionalized antibody functionalized clay in this case.
Okay. So the clay template itself is very stable. According to the literature, we did not study it ourselves. The etched clay is again still silica, so we would not expect it to degrade. But again, we've not tested it ourselves. Regarding the modified clay with the biomolecules, here there is a challenge of stability, no doubt. At the moment, we store them under -80, and they might, for dry state, they might need lyophilization. So we still don't have results for the stability of the whole system. But the clay as a template is not expected to degrade over time.
Okay. Thank you. Thank you very much. Do we have another question for Offer? All right. So I think we can close the session. I propose to thank. It's a virtual webinar. It's a webinar, and so it's virtual. But we can thank our speakers, our two speakers this morning. They are, remember, young scientists in their PhDs, and this is very amazing talks that we had with very interesting results. And it was a very exciting session, and I look forward to welcome new young speakers in the future. Thanks for uploading, Alexander. It's great. Thank you very much to both of you, and congratulations again for this great work.
Just before,
you're welcome. Before we stop, I have, if possible, Luca, let me know. I would have a short announcement to make eventually if I have a chance to share my screen, which I'm not sure how to do on Google Meet. Let me know. Maybe, yeah. I will tell you what it is about.
There is a square.
Yep.
And you can decide to share the screen or a window. Can you get it?
Where is that? Yeah, I'm sorry.
There is a rectangle with an arrow.
Yeah. I'm in this already.
And then you can choose whether to share the screen or a window of your application you're using, like PowerPoint or what else.
Okay, so I try to share the screen. Oh, now I have to put this on my screen. Not sure it will work. Fine. Yeah, no, let's ignore this. I should have prepared that. This is just an announcement of a coming meeting that will be in Belgium in November 9th and 10th. It's proposed by the French Society of Semiconductors and Oxides. It's exactly the same field that Netpore is in, and this is a combined event that we organize in Belgium in November. And it's quite small community. The French community is combining this with the Netpore event, and we have an abstract called Open right now, and we encourage young scientists to apply. That's why I make this announcement today, because we favor usually a student and postdoc for oral communication. So if you're interested, if you want more information, don't hesitate to email me, and I will send you the link of the website for it. And again, thank you very much for your participation, and thanks.
Maybe, Freddy,
Thank you,
we can publish. Maybe we can publish the link on the events page of the Netpore website. No. Ciao. Freddy, can you hear me? No.
Should we close the session? Yeah. Are you closing? Yes.
Can you hear me?
Sure.
Freddy, can you hear me?
Yes, I can.
Okay, so I was just to add that we could insert the link of this event in the page of the events of the Netpore website, maybe.
Yeah. I got it. It is already on the Netpore website in the page. Yeah, of the event. Yeah. So that's Thank you, Luca. Should we close the session?
So everybody can find it.
Yeah, people are gone, I guess. Yeah. Have a great day, all. Bye.
Okay.
Thank you. You too. Bye.
Bye-bye.
You're welcome, Mariana. Thank you all. Thanks for the great talk.
Thank you all. Bye-bye.
Bye-bye.
Bye-bye.